Hi, my name is Hunter LaRochelle,
And my name is Emma Cowie.
and today for our capstone we will be discussing the effects of urbanization on aquatic environments.
Specific land uses around aquatic environments greatly impacts water quality. Homes, businesses, farms, roads, industries, and other aspects of urbanization typically affect aquatic systems through surface runoff. Human activity of land use surrounding aquatic environments have accelerated rates in degradation of drinking water, fisheries, and other freshwater activities and parameters.
Because of these different factors that can affect water quality, we hypothesize that the species will vary from those found in urbanized rivers compared to those in non-urbanized, with a low population of species from these differing water qualities. This will be shown through lower dissolved oxygen levels when comparing the urban sites versus the non-urban sites.
To test our hypothesis, we deployed baited minnow traps each Friday to sample organisms. Each trap was baited with 1 cup of dog food and 1 piece of bread, as previous capstone projects have shown positive results with using this method of baiting. On Sunday, we collected traps to see if we had captured any organisms. If so, they were then identified, and then we also tested pH and dissolved oxygen levels. We tested 6 locations in eastern and western Massachusetts, 3 of which were in urban sites and 3 were in non-urban sites.
For the sampling locations, it was a mix between those found in western Massachusetts, in New Hampshire and Franklin County, and those found in eastern Massachusetts in the Bristol and the Plymouth County.
While hoping to capture more organisms during our sampling, we did capture limited species, but we did identify 5. In our eastern Mass samplings, 2 crawfish and a darter were captured. In our western Massachusetts sampling, 2 creek chubs were captured.
As of all collected species, were very tolerant species and can survive in a variety of water parameters. For example, the pH range for most survival and growth of creek chubs is between 6.0 and 9.0. The preferred levels are between 4.5 and 6 ppm for dissolved oxygen. As the Green River, where creek chub was captured, measured with the pH of 6.9 and dissolved oxygen of 7.11 milligrams per liter. This location was the ideal habitat for the creek chub.
The average pH of rivers in urban sites was 5.93 compared to the average of 5.65 at non-urban sites. We also recorded an average of 82.5% for dissolved oxygen in urban rivers, compared to the average of 83.6% at non-urban sites. This shows us while patterns show in each site, our p-values show these numbers are not a significant difference.
While very few organisms were caught overall, those caught did not tell us a significant difference in water quality. pH and dissolved oxygen levels varied by sampling site, but it was such an insignificant level shown by the p-value that it just didn't show a significant difference between urban and non-urban sites. We'd be interested in running this experiment again in the summer months, as the species are more active in the warmer months compared to the cooler months. So, in the end of the day, our hypothesis was not supported.